So, what are we going to be talking about today? We're going to be talking about probing the myeloma immune microenvironment one cell at a time. Now, if you are new, completely new, and wondering what the immune microenvironment even means, that's going to be discussed today. And then this is a test that does this is called single cell sequencing, and it's a sophisticated technology used to interrogate the bone marrow microenvironment. This allows researchers to do gene expression profiling of each single cell in the sample. And here to explain how it all works and how results from this technology can benefit the myeloma community is Dr. David Coffey. And as I mentioned, although I am a myeloma nerd, I work so hard to educate myself in all aspects of myeloma research. This is something that is new to me, and so I am excited to learn with you. It's my pleasure to introduce Dr. Coffey to you. He is a physician scientist at the Sylvester Comprehensive Cancer Center and the University of Miami. As a medical oncologist, he specializes in treating patients with multiple myeloma. His research is focused on understanding the interactions between myeloma tumor cells and the immune system. Leveraging his skills in computational biology, Dr. Coffey has developed analytical techniques to investigate the cells within the tumor microenvironment using single-cell gene expression profiling and DNA sequencing. He aims to understand how the immune system may fail to prevent myeloma progression and learn new ways to harness the immune system's power to eliminate the disease more effectively. Dr. Coffey, we're so excited to hear from you, and thank you for taking the time to be with us today. We're looking forward to your presentation. I think you're still muted, Dr. Coffey. We can't hear you.
Thank you so much for the introduction and for the invitation to speak today. I'm so pleased to be speaking with you all. I very much support what HealthTree is doing, and I'm excited to be a part of this education series. My name is David Coffey. I'm a physician scientist and assistant professor at Sylvester Comprehensive Cancer Center at the University of Miami. And today I'm going to be talking about my research probing the multiple myeloma immune microenvironment one cell at a time. My goal is to provide an educational talk, giving a high-level overview of what I do and why I do it. So while most of you may know, I'm sure there may be a few people in the audience who are unaware that multiple myeloma is a cancer of the immune system. Specifically, it is a cancer of plasma cells. Plasma cells are a type of B cell that originate in the bone marrow. Plasma cells normally function by secreting immunoglobulins, also known as antibodies. These are Y-shaped molecules that attach to bacteria, viruses, and mark them for their destruction by the immune system. Over one's lifespan, plasma cells gradually accumulate DNA mutations or changes in their DNA sequence. Some of these changes may be minor. Others might be more catastrophic, causing changes or rearrangements in entire chromosomes. Over time, these changes may give the cell a growth advantage, so it becomes a myeloma tumor cell. It continues to secrete the antibody protein, and since all the myeloma cells originate from the same clone, we call it a monoclonal protein. These monoclonal immunoglobulin antibody proteins can be detected in the blood and the urine of patients and can be used to track the disease progression over time. The antibodies can also attach to the kidneys, causing renal failure, or the myeloma cells can cause the osteoblasts and osteocyte bone cells to become destroyed so that patients develop these painful bone lesions that lead to fractures and also leach calcium into the blood that can cause damage in and of itself. Finally, the myeloma cells can overgrow the bone marrow microenvironment, causing the normal healthy cells to become reduced and leading to anemia. So the bone marrow microenvironment is a complex organ that exists within the bones, and it encompasses hundreds of different cell types. The bone marrow is located throughout the skeleton, primarily in big bones like the pelvis, the ribs, the sternum, the vertebrae, and the skull. The cells that occupy this space include blood-forming cells, cells that go on to be a part of the immune system, blood cells that carry oxygen throughout the body, and supportive stromal cells that give it a sense of structure, as well as bone cells like osteoblasts and osteocytes that help form the bone around it. And finally, it also contains some fat cells. Throughout the bone marrow microenvironment, there are blood vessels that give the cells that surround the bone marrow oxygen and nutrients so that they can grow. Importantly, there is increasing evidence to suggest that the cells within the bone marrow microenvironment play an important role in myeloma genesis. And we believe harnessing the power of these immune cells may help us more effectively treat the disease. The very first sign that the immune system could actually help eliminate multiple myeloma came in the early 1990s with the development of the allogeneic stem cell transplant. This is an older treatment in which patients whom are healthy donate bone marrow to patients with multiple myeloma. These cells are infused into the body and they replace the immune system of the original donor, of the original patient. And the reason that we do this is because the myeloma cells over time escape the immune system. They evade its abilities to detect them. And so the myeloma can outgrow the immune system. But when we put in another person's immune system, we're able to re-identify those hidden myeloma cells and can destroy them through a process known as graft versus myeloma. While this treatment was the first time we could see evidence of the immune system eliminating myeloma, it also came with serious risks. About a third of patients in clinical trials ended up dying within the first 100 days because of the severe toxicity of the treatment, as not only would the immune system recognize myeloma, but it would also recognize other organs in the body. Fortunately, we've come a long way since then, and we have now developed more sophisticated immunotherapy that have the ability to specifically target myeloma. The first of this was the immunomodulatory drugs. These are oral medications known as pomalidomide, lenalidomide, and thalidomide that activate the immune system in a generic way so that T cells, natural killer cells, and other cells that defend the body against cancer become reinvigorated to eliminate the cancer. However, these treatments are not specific to myeloma itself, and they also have broad side effects and toxicities because they affect other cell types in the body. We have since developed more specific immunotherapies that target proteins on the surface of myeloma. These include daratumumab and elotuzumab, which target CD38 and CS1, respectively, on the surface of myeloma. Once these antibodies bind, they elicit other cells, such as natural killer cells, to become activated, and they send death signals to the myeloma cells to eliminate them. While this treatment is highly specific for myeloma, unfortunately, it's not quite as potent as we would like it to be, and it often needs to be combined with other treatments to be successful. We have since developed even more sophisticated and more potent immunotherapies. The first of its kind was known as chimeric antigen receptor T cells, or CAR T cells. Here we take T cells from a patient's bloodstream, we genetically engineer them in a lab by inserting new DNA from a virus that then encodes on the surface of the T cell a brand new receptor called the CAR, or chimeric antigen receptor. This receptor has been specifically designed to recognize myeloma proteins on the surface of those cells. We then expand the CAR T cells in culture outside of the body and then reinfuse them into the patient. Those CAR T cells then go throughout the body, find the myeloma cells, and eliminate them. So we can think of this as a living drug. While this treatment has been very successful, it unfortunately has some toxicity. Specifically, it can cause cytokine release syndrome. This is when patients develop a flu-like illness due to overstimulation of the immune system. The other disadvantage of CAR T cells is that they are challenging to manufacture. So to overcome this, bispecific T cell engager therapy has recently been approved. This allows us to generate essentially the effect of a CAR T cell while it's inside of the body. So it's a linker protein that links together T cells by the CD3 protein on their surface with myeloma cells by attaching to a protein such as B cell maturation antigen, or BCMA. By linking T cells and myeloma cells together, the T cells become activated and again release death signals to the myeloma cell to eliminate them. And the advantage of this treatment is that it can be given as a subcutaneous shot, and it doesn't require gene modification therapy that can take weeks to do outside of the patient. So we were very excited about this type of treatment, and there's multiple variations of this coming down the pipeline. So over the past several years, we've seen great improvements in our immunotherapies, and we've seen results in patients who have failed all prior lines of treatment, indicating that it is an excellent type of treatment to give. But about 50% of patients within the first year of receiving the treatment do end up progressing. So it's not a cure-all. So the focus of my research is really to understand why some patients achieve an excellent remission and others do not with immunotherapy. And to accomplish this, I am using new technology to study the immune microenvironment where the treatment is working and other immune cells are involved, so we can understand aspects about why the treatment works or why it fails, and if we can identify in advance which patients are most likely to benefit. So let's talk a little bit in general about how research scientists study the immune system. So there are two general categories of immune research. The first is we do immune stimulation. So the goal here is to stimulate the immune system and see the effect that it has. And the other type of research is looking at immune detection. So here we're looking at the effects of immune stimulation using a variety of technologies. So let me go a little bit deeper. So in terms of immune stimulation, we can take immune cells out of a patient's body, we can put them in a culture dish, and then we can stimulate them with proteins, maybe a vaccine, or maybe it's a drug. And then we can see the effect that this has on the immune cells based on if they're dividing or if they're releasing proteins. All of those types of pieces of information tells us whether or not this is a stimulating agent or not. We could also do it in an animal model that has a competent immune system itself. So we may inject myeloma cells into a mouse model, for example, and then we can track over time whether the treatment we give those mice is effective by looking at the interaction between the immune cells and the tumor itself. Finally, of course, we do clinical trials. So once we know that the drug is safe and effective, we can test it in larger patients and we can see its effect in the human body. So how do we determine if a treatment is effective? How are we measuring the immune system itself? Well, there are three big categories. We could look at the tissue, we could look at this individual cells, which I'm going to get in more detail about, or we can look at the molecules made by the immune system, such as the proteins. So here are three example technologies that we've traditionally used for many years to do this. The first is immunohistochemistry. So this is commonly used if you undergo a bone marrow biopsy. So we take a core of that biopsy and we slice it into small planes. We can then apply an antibody that recognizes proteins on the surface of the cells in that slide. We then add a second antibody that will release a color if the first antibody is bound. We then look under the microscope and we can see if a cell is positive or negative for that stain. And that helps us infer what kind of cell it is. So if it's a bone marrow biopsy, we frequently look for plasma cells, the myeloma cell type. And so we can count under the microscope how many cells are plasma cells and how many are not. Another technology gets even more specific. What this does, it looks at one cell at a time. But for flow cytometry, the technology I'm illustrating here, you have to know in advance what cells you're looking for. And so you may add markers specific to B cells, or you might be interested in monocytes or other cell types. You add those markers, and then you pass them through the machine, and they go through a small capillary tube, one at a time, and a laser beam will pass through them, and the detector will detect whether or not that cell has the labels that you applied to it. And then you can calculate how many cells are in that sample of that specific type you're interested in. And lastly, we can look at the proteins. You might take some serum or plasma, the liquid portion of blood, and put it into a dish, and then add a primary antibody that will react if that protein you're interested in is present. So this allows us to quantify and detect whether or not a specific protein is in the blood. This is an example of how we do the COVID-19 assay. We can look for specific COVID-19 particles. Now, new technology has emerged in 2009 that allows us to do even more powerful interrogations of the immune system. And this is called single-cell sequencing. And to give you an idea of the impact this is having on science, I've plotted for you here the number of research publications introduced each year since the technology came out. As you can see, it's been growing exponentially. And last year alone, there was 5,000 research papers that utilized single-cell sequencing in their studies. Before I define what single-cell sequencing means, let me make sure everyone's on the same page when we talk about genes and DNA and what that all means. So genes are regions of the DNA that encode a specific kind of protein. And the way that the protein is made is through an intermediate molecule called a messenger RNA. So the DNA gene gets transcribed into a short piece of messenger RNA specific for one kind of gene and one kind of protein. It then gets translated into an amino acid sequence, like a chain of pearls, that then fold into three-dimensional space to create a unique protein. And ultimately, it's the protein that does all the work inside of the cell. And so if we think of it as an analogy, the DNA would be like the blueprints, the messenger RNA would be the construction workers, and the protein would be the final building that is being constructed. So how can we utilize this knowledge in single-cell sequencing? Well, within each cell, if we were to take it apart, we would see lots of messenger RNA molecules. And if we counted each molecule that corresponds to a specific gene, we would find that every cell type has a unique gene expression signature. We can use that information then to identify in a non-biased way which cells inside of a sample belong to which cell type. And so here's an example showing a clustering diagram where each dot represents a cell, and above it I show an example of a gene expression signature. So these are, each bar represents a different gene. You can see some are high and some are low, but all the cells in this particular cluster will harbor that same exact signature. And the important part about this technology is you don't have to know what you're looking for. You can just take all the cells in the sample, sequence them all, and then go back and look and see what you found. So it's an unbiased way to do research. So how exactly do we do this? As you can imagine, single cells are pretty small. And so are we using scalpels and opening them up and counting molecules? The answer is no. Instead, what we do is we take a sample of bone marrow or blood, we pass it into a machine that causes the cells to pass through a microfluidic circuit, one cell at a time, in single file fashion. They then meet up with a gel bead. This gel bead is important because it contains all the chemistry necessary for the sequencing reaction, but it doesn't release it quite yet. It waits until the gel bead and the cell come together in a single oil droplet. The oil droplet should only have one bead and one cell. Once that happens, then the gel bead will release its contents, And that contains all of the necessary enzymes and molecules that we use to do DNA sequencing. So the steps in this process are, first, it takes all the RNA molecules and converts them into a complementary, more stable DNA molecule. And then every oil droplet will have its own set of barcodes. And those barcodes will label each of the DNA molecules. So all the barcodes, for example, that are red will belong to the red cell. All the barcodes that are purple in this example would belong to the purple cell. Now we can break open those oil droplets and sequence all of the molecules. And we can use sophisticated software relying heavily on machine learning to be able to cluster the cells according to their like gene expression profile. So let me show you some real data. So here I'm showing you a clustering diagram of bone marrow cells from multiple patients with multiple myeloma. And as you can see, it forms this really complicated structure. Each dot here represents a single cell. And when they group together, they form a cluster of cells that have like expression. That is, they share the same signature. We can then look into a database of previously annotated cells to see which cell type they most likely are like. And so by doing so, we can very rapidly determine all of the cell types inside of a sample and infer what they're doing. So not only can we give it a label, but we can also get an idea of whether the cell is an activated cell or if it's been suppressed in some way. We can get all that kind of detail from a single cell. One thing I'm going to point out to you that was really profound to me when I first saw this was this plasma cell cluster. If you can see, it almost looks like a starburst in this figure, this orange starburst. This is the plasma cells. And unlike the other cells that are more uniform and discrete, this is actually composed of multiple subclusters. And so when I labeled each cell according to the patient and time point that it came from, I found that each subcluster actually belongs to a different patient. And so what this means is, unlike T cells or myeloid cells or other types of cells that have similar gene expression profiles, each individual myeloma patient cells actually has its own unique signature. This means no two patients are identical in terms of their myeloma. And this is really important for us to recognize. We can't expect the same treatment given to this two different patients to have the exact same outcome. It actually means that each patient may have their own unique response because of their differences in their gene expression profile, all caused by differences in the DNA mutations within that cell. So let's talk about some more detail and some exciting publications that we have coming out this year. So one study that we did a few years ago, and we're now finalizing and publishing this year, was to look at single-cell sequencing of the blood of patients with newly diagnosed multiple myeloma while they were undergoing maintenance therapy. So all the patients in this study were being treated with the same drug, lenalidomide. Now, the thing that we studied was patients who had sustained MRD negativity compared to those who did not. If you're not familiar with MRD negativity means no detectable myeloma cells using the best technology to date. So we compared those patients, essentially those with the best outcomes, to those who did not achieve that. And there were a few really interesting findings. Those patients with sustained MRD negativity tended to have more helper T cells. These are the kinds of T cells that help the other immune cells mount a response against the tumor. And so it wasn't necessary that they had to have the cells themselves that interacted with the tumor, but the helper cells, the intermediate cells, turned out to be really important. Second, we saw a reduction in regulatory T cells. These are suppressive cells that put the brakes on the immune system. They're usually there to prevent autoimmune disease, but we actually saw them in patients at higher levels who did not sustain MRD negativity. So we see reduction in those as being critical for long-term outcomes. And the final thing that I will point out that we saw was that patients who had sustained MRD negativity had a more similar immune system to healthy individuals. This indicates to me that patients undergoing intensive therapy have a good chance of achieving a healthy immune system at the end. Or the upside of that is maybe you have to have a healthy immune system in order to achieve a long-term remission. Either way, a healthy immune system is still achievable even after intensive therapy, including prior stem cell transplants. The next study I'll point out was looking at patients also with newly diagnosed myeloma, but this time receiving not maintenance therapy, but the induction therapy. These are the 4 drugs that we usually give patients at the beginning of their treatment: daratumumab, carfilzomib, Revlimid, and dexamethasone. We did the same structured study as the last one. We compared sustained MRD negative to non-sustained MRD negativity. And what we found is that the response depended on natural killer cells as well as monocytes to be in high levels to support the function of daratumumab. So the daratumumab antibody doesn't work on its own. It needs the help of natural killer cells and monocytes to be able to work. And we saw strong evidence to support this in this particular study. So what this means is that you need a healthy immune system in order to make your drugs work. And so that was the primary outcome from this aspect of the study. So where are we going to go into the future? I think there's a lot of work that needs to be done to improve single cell sequencing. It's still in its infancy, only been out for 20 years, which may not seem like, 10 years, which may seem like a long time, but there's still a lot of advancements that need to be made. The first is that we're only able to detect about 10% of the cells inside of a cell at one particular time. So there are 25,000 protein encoding genes, and we can only detect about 2,500. I'd love to see that increase over the upcoming years. The next thing is it's really expensive. It costs about $2,000 a sample. And if you have a study of 100 patients from a big clinical trial, sometimes it's just not feasible to look at every individual. But I think it really is important that we sample every patient in these studies because everyone has a unique response, and we need to know why that is the case and identify which treatments are going to be most helpful for which patients. It's also just research use only. So if anyone's watching this and thinking, when can I get my immune system profiled? We're not quite ready for prime time. We use it to study mechanisms of the disease and understand how we can improve treatment. I think in the very near future, we'll be using it to predict treatment outcome and inform decisions about treatment. And at that point in time, I think it will be more widely used. So I'd like to thank all of my co-investigators at Sylvester Comprehensive Cancer Center. I'd like to thank Ola Landgren, our leader of the Myeloma Institute here. He's grown our program to be of great size, and we continue to grow every day. And of course, I'd also like to acknowledge all the patients who are involved in our studies to make this research possible. If you are interested in participating in one of our research studies, some of which include immune profiling, feel free to reach out to our patient care coordinator, Ms. Tia Montgomery. Or if you'd like to just read about our consents that are available, please reach out to Michelle Armagen, who is our research manager, and these are the email addresses. Thank you so much for your attention, and I look forward to hearing from you regarding your questions.
Thank you, Dr. Coffey. That was amazing. I love how you started out really at the beginning, and then you were able to show us really the future of where this could go. Thank you for the work that you've put into this as well. I know that this is a personal project for you and something that you've been working on for a long time. How does it feel for you to have it now available for research use after all the years of developing this?
I was very fortunate to work with some companies on the development of single cell sequencing. At that time, believe it or not, we were pipetting one cell at a time into a dish. And each dish we would do the reaction. Now we have a nice machine. You can just drop in your sample and it will do it for you. So we've come a long way just in the past 5-10 years. So for me, it's really gratifying to see that this technology is being widely used and it's helping us understand the disease in greater detail.
I completely agree. As I said, I really appreciate the slide that shows where you're going in the future. I mean, this is the $1 million question, right? Because of this accelerated 5 to 10 years that we've seen so much progress, do you imagine that same acceleration to continue in the next 5 years for us to be able to see that progress that we're hoping for?
Yes, I absolutely do. And just last week, I attended a talk about some new technology coming down the pipeline. Now we're going to be able to not only sequence all of the RNA molecules, but also all of the DNA molecules that allows us to determine if there are mutations, if there are translocations, to
Mm.
really interrogate the entire genome. And I think combining the two is going to be really powerful and allow us to associate what changes in the DNA are influencing the expression of the genes.
I completely agree. Follow-up question with that. As you know, as I think a lot of us know, African Americans are 2 to 3 times more likely to develop multiple myeloma. Do you see this test as a way that we can figure out why that's happening, or also how specifically that population is responding? As we know, not everybody is the same, but there might be some similarities.
I think that's a really important question. And I thank the person for asking that. That's something that we're very interested here in Miami to try and answer. We have a larger African American population in Miami than in other locations throughout the United States. So we're proud of the fact that we do enroll a number of African Americans in our study so we can answer those kinds of research questions. I would also say that there might be environmental aspects that are
Mm.
different in certain populations, ethnicities, and others that influence the immune system. There's some people, for example, have been studying the microbiome. These are the bacteria that live in us and around us, some of which are helpful to us. And that influences our immune system. And that may relate to different ethnicities based off of where they came from, where they live. And I think trying to tease all that out is a super important question. And we look forward to addressing that in the near future.
Definitely. Very exciting progress that's being made. And again, just thank you for your work. One other subgroup question in relating to this test. As we know, Agent Orange is one of the known connections between myeloma and, or like diagnosis of myeloma. It would be really interesting, and I'd love to hear your thoughts on using this test to see how do those who have that connection with Agent Orange respond to certain medications versus others who don't really know the connection of where their myeloma originated.
Yeah, that's also a great question. So we know that Agent Orange has some mutagenic effects on the DNA itself and can be the source of origin for why a myeloma cell developed mutations and eventually became a tumor over time. But I would hypothesize that maybe it also had impact on the immune system.
Mm.
And maybe it made the immune system weaker. Maybe it influenced the immune system to stop fighting the cancer cells it was normally designed to do. And so it would be great to do some studies looking at patients with Agent Orange exposure and not to address that question. At this moment, we haven't got a specific study to do so, but I love these ideas because it inspires me to do these studies.
Well, I just love that there's potential for these studies because of the technology that's being created. It's just really exciting that it's a deeper look into these questions that we've had so that we're able to find answers. Lots of great
Exactly.
questions from our audience. I'll start here with Doug's. Are you doing any work on T cell exhaustion? What causes it? And if it can be prevented?
Yeah, that is also a good question because we do a lot of research on T cell exhaustion. One of the challenge has been a uniform definition using a gene expression signature to define exhaustion. And so what we've done is to try and identify through lots of prior literature and other experiments, what are those genes that definitely indicate that this cell has become exhausted and is no longer functional. And in doing so, we have identified higher levels of exhausted T cells in patients with myeloma Now, for those who don't know what that means, so over time, a T cell will become repetitively stimulated, whether it be by an infection or by a tumor. And that repetitive stimulation often does an opposite effect and tells it to turn off. And when it turns off, it no longer recognizes the tumor or the infection. And so that's problematic. So we do need to find ways to reinvigorate them. And some drugs have been evaluated. These are called checkpoint inhibitors. And unfortunately, the first trials did not show they were very successful, but people haven't given up. There are more trials ongoing. And I think we need probably some different approaches, maybe not the same kind of checkpoint inhibitors we use in breast cancer or solid tumor malignancy, but the kind of checkpoint inhibitors we need to design should be specific to myeloma. So it's a very important research question that we're actively looking at.
Wonderful, thank you. Let's get into the specifics of clinical trials that's using these tests. One person here is wondering, as a patient at Sylvester, can you get this test performed as a part of a clinical trial?
Yes. So I can tell you 2 trials that we are actively doing immune profiling on. The first is for patients with relapsed multiple myeloma who would be eligible for the drug teclistamab. This is the BITE therapy I talked about. If you haven't received this treatment yet, and if it's appropriate for you, you should be considered for this trial. You can meet with one of our physicians here that are enrolling patients, and we would profile your immune system as a part of this treatment. The second would be something called the TRANSFORM study. This is a very large-scale study we're doing in Miami, and this is for patients with MGUS or smoldering myeloma. So if you haven't developed myeloma yet, this study is profiling the immune system as well as the genome in patients to see if we can predict which patients are most likely to progress. Because right now it's a watch and wait game. And for some, that's a real nail-biting experience. Wouldn't you like to have a test that could tell you with great accuracy what your likelihood of progressing is? Well, that's the aim of this study. So if either of those are interesting to you, feel free to reach out to us.
That's very exciting. Do you have to go to Sylvester for those? They're not off-site clinical trials?
These sites are all being conducted on site, but you don't have to necessarily reside in Miami. So for patients, for example, in the TRANSFORM study, they only have to have a sample once a year. So you could live outside of Florida and come visit us once a year, have the testing done, and then leave. Now, I will say that the testing isn't immediately available because it's research use only. It does need to be analyzed. We do have a lot of people looking at it, and it can take time to get all the information together. So unlike a clinical test, we get the result in a week. This may take quite a bit longer.
Yeah, understood. It's making progress though, which is the exciting part.
Exactly.
A follow-up question to those trial qualifications. That first trial you mentioned, do non-secretory patients qualify, or is that something that would inhibit them from enrolling?
No, non-secretory patients should be eligible.
Thank you. Paul asks a great question. Will AI provide faster results? And if you could just expand on how this development of technology that we're seeing can aid in the research of myeloma. That's a big question.
I love that question, Paula. I'm a huge advocate of some of the benefits that I think AI has to offer us. Of course, we hear about some of the risks, but in terms of the benefits, I think the scientific field and the medical field has a lot to potentially benefit from. For example, there are dozens of millions of publications generated every year in the sciences. Like I mentioned, 5,000 just last year on single cell sequencing. It's very hard for me to keep up with 5,000 scientific papers. But I think AI has the ability to sort of synthesize that knowledge. And so I think in the very near future, scientists may first go to the AI agent and ask their opinion, if we can call it there, what the next research question may be based off of all the prior literature. Also, I think AI is helping us to analyze the data. You can imagine if you were looking at 1,000,000 cells and you're looking at thousands of genes per cell, it's just a huge data set, very hard for my brain to fully process. And I think AI can do sometimes a better job at that and find patterns that I didn't recognize before. So I think AI is going to help us in discovery, and I think it's going to help us in analyzing complex data sets.
Awesome answer. Thank you. Let's go into a little bit of while we're waiting for this amazing test to be widely available, what else can patients do for their immune system since you are interested in the microenvironment of their immune system? One
Yes.
of the questions here was about nutrition and physical activity. In your investigations, have you found that those have a real impact on the immune system?
Yes. So I haven't personally analyzed data or done research studies looking at exercise and immune system, but I have read plenty of research to suggest that there is a strong correlation. So if someone were to come to me and say, Dr. Coffey, how can I improve my immune health? I would say these things. I would say first, you have to have a healthy diet. Low in saturated fats, red meats, high in fruits and vegetables, grains, fiber. Second, you've got to have some exercise. And it doesn't mean you have to train for a marathon, just a daily walk at least per day. But if you're able to do more, then push yourself and keep track of your steps. Try and walk more today than you did yesterday. The third thing is, I would say, get good sleep. Sleep is really important for our health. And unfortunately, some of the treatments we prescribe can make it worse. I acknowledge that. But if you can get good sleep, that is really important for our immune system. And the last thing I would say is try and reduce stress. And I know that's easier said than done, but there are things that we can do, mindfulness training, meditation, yoga, being with people who care about us, those things can help reduce stress. And I think everyone should try and achieve those goals. Notice I didn't mention a magic pill. We do not yet have a treatment that can per se enhance one's immune system. So there's immune booster tablets that you buy at a health food store or supplements like vitamin C. I'm not a strong advocate for because I don't think that they can do enough to try and improve our immune system.
Thank you very much. Yeah, there was a question here specifically about mistletoe, and I know other people are curious about which supplements can be the most useful, and so I appreciate that insight. Um, Terry's wondering when we were talking about MRD negativity, what does sustained MRD negativity mean in clinical trials?
That's a great question. So first came MRD negativity, and that means at any point in time, you would get a bone marrow biopsy, and we would look using the best technology to see if there were any myeloma cells. And at this time, the best technology can detect 1 in a million myeloma cells. So very sensitive. However, we found that just a single measurement was not as predictive of a long remission as having multiple measurements sustained over at least 1 year. So if you have a measurement in January and then you have it again in December, and both times it's negative, we would say you've achieved sustained MRD negativity, and that really predicts the best outcome for patients.
Thank you. A follow-up question with that from Doug. NK or natural killer cell depletion is important for sustained MRD negativity. How does this fit with the need for NK cell involvement with DARA efficacy?
I think if I said that, I'm sorry, I flipped it. It should be high NK cells are necessary for daratumumab efficacy.
Okay.
So natural killer cells have a receptor on their surface that binds to the tail end of a daratumumab molecule. And so when the daratumumab is affixed to a cell, NK cells will bind those daratumumab molecules and then become activated. And that's when they start to release their signals to kill the myeloma cell. So we confirm that even though it was suspected through in vitro testing, we confirmed that in patients receiving the daratumumab treatment.
Thank you for that clarification. Um, I'm just going to ask you some rapid fire questions about other immunotherapy treatments, if you don't mind, considering this is the
Please.
immunotherapy chapter. Carol's wondering about CAR T and bispecifics eventually replacing the stem cell transplant in the future. Do you see that? I mean, that's a very commonly asked question, and we don't know the answer yet. But what's your professional opinion?
I think that we'll always have the opportunity to do a stem cell transplant for patients whom fail CAR T, who fail BiTE therapy. But I think what's going to happen is not that we're going to replace it, but it's going to be pushed to further and further down the line so that we focus first on the less intense, less side effect type therapy. So hopefully patients will undergo CAR T cell maybe in their first relapse, as opposed, or maybe at their diagnosis, as opposed to waiting to their fifth relapse. And the reason I think that's really important we sequence it that way is if we did the transplant first and then we gave the CAR, we're going to be using patients' immune system to generate the CAR after they've been beat up by the transplant. So I would love to give the CAR as early in therapy as possible when the T cells are the healthiest. So to summarize, I don't think it's going to replace it. I think it's going to kick the transplant can down the road too much further.
Yeah, very well said, because the stem cell transplant works for the majority of myeloma patients. So why get rid of something that is efficacious when it could be used in the myeloma arsenal? Okay, I love that answer. Thank you.
Exactly.
Another question: what's the status of development, if you know, of personalized anti-multiple myeloma neoantigen vaccines?
So that has been something that people have been working on for a long time. And it's not only in myeloma, but it's other solid tumor malignancies. I'm very excited by the concept of these vaccines to explain it to other people. So neoantigens are proteins that are displayed by myeloma cells that originate from a DNA mutation inside of their nuclei. So only myeloma cells should have these specific antigens. But unfortunately, over time, the immune system stops looking at them. So if we could vaccinate the immune system with more neoantigens, maybe we could overwhelm the immune system to take another look, let's say, and then go after the tumor cells. I'm not aware of any of the latest in terms of the efficacy in the past couple of years, but I know, like for example, with the MAGE-A neoantigen vaccine, there were some promising results. I think just more work needs to be done before it reaches mainstream and FDA approval.
Thank you. And then what are your thoughts on a tumor infiltrating lymphocyte therapy approach to treating multiple myeloma?
So tumor infiltrating lymphocytes, also known as TILs, are when you take the T cells out of the tumor and you expand them in the lab and then you reinfuse them with the thinking that those T cells in the tumor are going to have the most reactivity to the tumor. But in myeloma, it's a lot harder, right? Because myeloma is distributed throughout the bone marrow, sometimes throughout the blood. And it's not like a solid tumor cancer where it's clustered into these small tumors that you can then excise and extract the T cells. So I'm not sure that TIL therapy is as applicable to myeloma as it is to the solid tumor cancers.
Thank you. Karen wanted to clarify, did you say that CAR T is a live vaccine, which it's not, correct? Okay.
No, it's not a vaccine. It's genetically modifying one's own T cells. We do use a virus in that process of genetically modifying them, but in the end, it's not a vaccine. It's a stimulation of the immune system.
Yeah. Speaking of, earlier we were talking about maybe other cancers, other diseases. Do you see this tech helping treat MDS in the future?
You mean single cell sequencing?
Yes, sorry.
MDS, there's been a lot of single cell sequencing studies in MDS and AML. In fact, they are one of the leading groups in single cell sequencing. Um, I think that it definitely has contributed a lot to the knowledge about the disease and is helping to generate better therapies. So a resounding yes.
That's very exciting. Thank you. Okay, there are lots of questions and we're not going to get to all of them. I already apologize to our audience. I'm not purposely ignoring any questions. Carol's wondering, can you repair the gene? Is there gene slicing and repair in the future? I also get this question often.
The problem with that is doing gene therapy inside of the body would affect all of the cells in the body. In myeloma, only the myeloma cell's DNA is damaged. And so it's very challenging to target the correction of the damage in the myeloma cell itself. In fact, it's just a lot easier to kill it and to eliminate it rather than try and rehabilitate it, because the body has the ability to make new healthy plasma cells, and it's doing so all the time. It's just you have a rogue group of plasma cells that need to be eliminated.
Thank you. Do patients with autoimmune diseases have different outcomes, given that their immune systems are highly active? Are you aware of this
Well,
in the early research?
yeah, I like this question. There are some research to suggest there might be a correlation between patients with autoimmune disease and the development of myeloma. I have seen a number of people in my own clinic who have had, for example, scleroderma, rheumatoid arthritis, or lupus, and a myeloma diagnosis. As a side note, many of those patients actually experience relief of their autoimmune diseases while they're being treated with myeloma therapy, because the myeloma therapy is often targeting the other immune cells that may be causing the autoimmune disease. I can't say, though, that I've seen greater severity or different outcomes in patients with autoimmune disease and myeloma. Maybe that research has been done. I just don't know the answer to it.
Thank you. Well, I'll try to answer a couple more questions because we have a couple more minutes. Let's see. Oh, it looks like I was mistaken that mistletoe is not a supplement. It's a medicine that one needs a prescription for. Have you used mistletoe ever in treating multiple myeloma or have heard of any research?
I have to be honest, I'm not familiar with using mistletoe in myeloma, but I'm open to learning more about it.
Thank you. Vicky's wondering, has anyone studied long-term myeloma survivors to see what their cells might have in common? Or is that something that you would like to do maybe in the future?
Yeah, that's a really good question. I would especially like to do that in the context of the immune system. I'm not familiar at the top of my head of a study that specifically looked at that. We are all very familiar with patients who have had the disease for more than a decade, perhaps maybe even cured. And we want to know why those had such a good outcome. If we had enough of those patients come to us and say, here, test my blood, we would do so in a heartbeat.
Well, I'm glad you said that, because now I'm going to contact our research team here at HealthTree and say, you know what, let's get this, we have the records of so many of these awesome
Yeah,
long-term survivors. Let's do something about it. We'll be
I would love
contacting
to
you, Dr.
collaborate.
Coffey.
I
Let's
would love to
collaborate.
collaborate.
Such
Yeah,
a good
yeah.
idea.
Maybe they could do a blood sample locally and then have it shipped to us, and that way we can reach as many people as possible, not just those in our area.
Yeah, definitely. Okay, I'll have our research team contact you. That's so exciting. All right. One of the other suggestions here, our audience is so well educated, using the technique investigating comorbidities and their reactions to multiple myeloma treatment. I mean, that would be fascinating to see, those with high hypertension, those with diabetes, how are they responding differently?
I agree. I think that we need to think about the patient's total medical health when we are evaluating the efficacy of these treatments. Because I agree with you that there are comorbidities that influence treatment outcomes that we just haven't yet appreciated. We're trying to build an extensive clinical database of our patients here in Miami so that we can identify those trends. Another way that's research like that we're trying to take part in is there's an NIH funded program called All of Us. And what it is participants can give a blood sample and complete online surveys about their health. And all of that information is protected and sent to researchers so that we can look at factors like hypertension and see if they are related to other treatment outcomes in myeloma. So I think that's an example of a study if you yourself are interested in could participate and help us learn more.
Wonderful. Thank you, Dr. Coffey. I just wanted to read here. Please thank Dr. Coffey for his very precise and clear information. You're an excellent presenter and just a genius using his genius for good. So thank you for what you're doing. We really do appreciate it. I'll give you these last minutes to give any closing remarks that you'd like to give.
Thank you so much for that kind remark. I really enjoy educating and teaching, and I have a passion for making a difference for myeloma patients. And that's why I became a researcher, so that I could push the limits and the boundaries on what we're doing. And I hope that I fulfill my goal of curing myeloma in all of yours lifetime, not just mine. So thank you so much for inviting me. It's great to be here, and I look forward to being back in the future.
Thank you, Dr. Coffey. Beautifully said. And thank you again for spending time with us today. We're going to let him go, but to our audience, we're just going to continue with a couple of outro announcements. Tonight, we have another event, nutrition and GI side effects. As we know, chronic diarrhea, constipation, nausea, those can affect our day-to-day life. So we are having a dietitian come talk to us about how we can manage those, especially with all the different medications and stuff that exist within us. And then on the 22nd, which is tomorrow, we'll have our Black Myeloma Health chapter, and we're going to be doing a live exercise demo with Vanya. And then on the 23rd, which is Friday, we normally don't have events on Friday, but doctor's availability only allowed him to come on Friday, and I was really wanting him to come speak to us. It's Dr. Ollie. He's going to talk to us about if MRD, or minimal residual disease status, should be used as a good endpoint in clinical trial drug approval, excuse me. The link to sign up for any of those events and even more events I didn't mention is found at the bottom of the slide and will be included in our follow-up email. Another thank you to Amgen, AbbVie, Adaptive Biotechnologies, Janssen Oncology, Genentech, and Bristol Myers Squibb for their sponsorship. And thank you to each of you for helping us build this myeloma community. I'm grateful to each of you and your desire to educate yourselves, and hope you have a great rest of your day. Take care, everyone. Bye-bye.